after. Today we will talk about beta membrane prediction. So last week, Tuesday, we talked about, last week we talked about the alpha helical membrane proteins. Now some proteins go with helices that are orthogonal to the membrane, through the lipid bilayer. Others do it with beta strands. The beta strands, if you imagine this to be the lipid bilayer, so the heads groups in the middle, so it runs around the cell like this. Then the beta strand membrane proteins essentially sit on a 45-degree angle where the strands are formed by my fingers, hydrogen bonds between the fingers, and they wrap around it. So you see that here, and you see that essentially they form these pores. When you look at it from the top, you essentially see a hole in the cell. Here are a few examples. The few examples essentially differ in the way they are inserted. You see they differ in the width of the barrel, essentially by the number of transmembrane strands that span it, which changes the width, and they differ a little bit by the way these outside regions, extracellular inside regions look. When we looked at, so this was from Henry here, 30 years ago. The list is not much bigger today. When we looked at the entire set of 3D non-membrane proteins, or beta barrel membrane proteins, for which we know high resolution structures that are sequence unique. So this was the entire set that was available. Again, as I said today, the set is not much larger. As you remember, the problem is that in the crystal we don't see the lipid, meaning where exactly this cuff here, blue and red, are the hydrophobic cuffs, are the membranes. Bless you. Since you don't see them in the crystal structure, you guess them. The word hydrophobic cuff already indicates how that is guessed, simply by seeing that there's a set of hydrophobic turning into non-hydrophobic residues at the interface. And that is where you essentially assume the membrane to lie. Again, experimentally you don't see it, you don't see lipids. So this is a simulation. Now once you have this, and initially Henry really did that, he did that before these methods existed, that look at the cuff, he did it visually, and then began to label what is a strand that goes up. So up strand is simply that goes outside. What is a strand that goes down? So it goes inside. There is a loop inside, and there's a strand, a loop at the outside, right? And those are the states. So you take your structures and you assign for every single residue which of those 4 states is it in. Or there could be a 5th state, which is none of the above, so to speak. So you could be in a globular domain inside. That could also go to 6th state. But initially we really looked at a model that has these 4 states. And so again, in terms of following through running through the model, you have these alternating inside, up and down strands. Then you have the loops inside and the loop outside, and you build a model. Now, this model, if you look at the few structures, makes sense. How can you figure out whether the model is right? What would you do? So the model, essentially, to repeat what I call the model here, is essentially you have 4 major states. There is something that models the inside loop, there's something that models the outside loop, and there is an upside and a down, an up and a down strand. This implies that in fact there's a difference between these two, right? You put that in a model because you argue that yes, this is a loop, this is a loop, but they differ because they're on different sides of the membrane. Yes, this is a strand and this is a strand, but they differ because one goes up and one goes down. How can you see whether that model makes sense?
Test it with data, when you know what is right.
So when you're right, Simon, more explicitly, how can you test it with the data? So I give you 15 proteins. In these 15 proteins you have those 4 states. What will you do next?
Apply the model and
So you predict. I want to go a step before you go into the prediction, because developing a prediction method typically is not the kind of thing that you can do within a day. So what can you do in a day in order to really implement this model, do the priors right and do everything right, will maybe it takes you 2 weeks. When you invest something for 2 weeks, and if you can spend an afternoon on thinking or on gathering some data that will indicate that the 2 weeks are well spent or not. So you should do that as a first step. What would be your first step in this, Franziska? Did you have a question?
I was trying to say that if you want to see if the model makes sense, you have to compare with a different model that's simpler.
Yes.
Because if you make it with these 2 strands, then and you say you use them because they make sense, then it would have to be better than using 1 strand.
That is very true what you say. But that means, so ultimately what Simon says is predict and see how well it works. And then you say, when we do that, we have to also see whether there is a difference between the model and a simpler model. And that's all right. But whether the model works at all, or is that the promising direction? What would be the first step you would do? I'll give you a set of residues labeled in these 4 states. What will you look at in the first half hour? Or what will be the first thing to plot? What would you plot first? Is there any evidence in the structures that I gave you for supporting that there is a model with 4 states? How would you answer that question? How would you explore that question? What would you plot? Maybe it's too simple. Maybe it's one of the kinds of questions that are too simple to ask. The simplest thing is, I give you residues in 4 states. You simply count how many amino acids are in each of these states, right? And those are in fact here the 4 states, and the 4 states come here with the propensity of a residue at a particular position in that state. Okay? Now, the height of the letter is proportional to the degree of observing that letter. And what you immediately see, and it takes you certainly not a minute, but this is something that you see right now, is that they differ. Maybe you argue the differences are very subtle, but immediately it jumps at your eyes that there are in fact 4 different plots here. So whatever position you pick, whatever residue you pick, or whatever amino acid you pick, you will see that the others differ. So whether then the prediction method can pick that up, that was Simon's idea. So how well do you do in actually picking this up? Whether that is better than merging 2, because maybe in picking it up you make a mistake, is now the next question. But clearly the difference here between those 4 columns shows you that to invest the next 2 weeks into seeing whether there is a difference makes sense, because there is reason to assume that you actually have evidence for 4 states. If you had had 2 different ones here, or these 2, and they would have been identical, or much more similar than they are, you would have said, well, okay, maybe my prediction method still can pick up a signal, but at least on the first order signal here, they are similar, so maybe it's better to model them as both strands is 1 state, or both loops are 1 state, or whatever is similar here, right? But there's evidence for 4 different states. And in fact, since you have that evidence, it's not only that you sort of prevented 2 weeks of doing the wrong thing, actually you can even use that now as priors. Because the idea then is to put this into a hidden Markov model that models exactly these transitions. Yeah, maybe I should have started later, I'm sorry. I should have acknowledged the subway issues more than just jokingly. Please accept my apology for that. For those of you who come late, we are talking, let me just go back quickly, we're talking now about the prediction of beta membrane proteins. So this is the second class of proteins that go through the membrane with beta strands. They form pores, and we're trying to model it by a simple Markov model that distinguishes between strands that go up outside, strands that go down inside. Outside and inside is with respect to the cell, so outside the cell, inside the cell, and loops that are on the outside, loops that are in the inside. We assume that there are 4 different states, modeled by 4 different states in the hidden Markov model, and we have evidence we have accumulated evidence that in fact here's the letter height proportional to how often that letter is used in one of these 4 states. The 4 logos, sequence logos that you see differ visually. Let's not discuss in detail how much they differ. They differ. And since they do differ, you have evidence that you can really distinguish 4 different states. And now the next step really is to implement that. Implement that. The first step for a hidden Markov model always is to have priors. So the 0 assumption probability. So when you essentially begin to train the model, you begin with the background probability, and these are the priors for the background probability. So how often do you observe a particular amino acid in one of these 4 states? That's the simplest prior. And then this is more detailed prior, because this does not only tell you how often do you have a W in that state, but how often you have a W at that particular position in that state. So that's a prior in a more detailed level. So you put these high resolution structures in there, and then you get the performance shown here, where this is the raw number matrix. Number of residues is what the number is. It's the confusion matrix. In that direction is predicted, in that direction is observed. Diagonal means you predict it right. Predict right for upstrand, downstrand, inside loop, and outside loop. And you see that the diagonals are the highest numbers, so essentially you can, at an amazing level of success, distinguish between upstrand and downstrand. So in fact, if this is in some sense the confusion between upstrand and downstrand is not the most frequent mistake. If you make a mistake, you confuse the upstrand more often with the loop outside, right? And this sort of most likely comes from the fact that some of the residues at the end, because you don't see the lipid, you don't exactly know when the beta strand ends. So for some of these upstrand residues, you may call them loop residues. Not only in the prediction, but also in the labeling. Okay? So this confusion here, in some sense, is one that has to do with the unclarity of the data. I'm not saying that these are correct predictions. I'm only saying that there's some gray area that explains why the biggest mistake that you do in an upstrand prediction is the one for the outer loop. Because most likely that is where you're mostly running into. But again, I point out that this is clear, so you can clearly distinguish between upstrand and downstrand, and you can also clearly distinguish between loops that are at the outside and the inside. And again, for the beta membrane proteins, these loops tend to be relatively short. Let me pull up a set of structures again to show this point. You see many of these regions here, loop really is a loop, as in something that goes around, not as in something here. On that side here, the loop is not really a loop. There you run into a real globular structure, into a real protein again, right? This is not a loop. This is just a set of beta strands. We call that loop because it's at the outside. But in many of these cases, you see that they are relatively short, so we are really talking about loops. And again, the loops inside and outside in the prediction model are clearly distinguished. So the confusion here is relatively little. This and that, and that, and that is very little. Okay, this is a remarkable performance. But one issue, this is a performance on a level of per residue. So we are asking, what if I know my protein is a beta barrel protein? Where are the strands? I do very well. But can I discover beta strand proteins? For that, what I have to do is I have to project this core onto an entire protein value. And that is not that trivial. In this particular case, we sort of summed it up over the model. It's easy to sum it up, but to sum it up in a way where it makes sense is not that easy. So it gives a bit score here. You see 2 lines, the accuracy. The bit score increases the information content, so the reliability of the prediction, so to speak, for the entire protein increases. On the right-hand side, there are cases where you're more certain that they, in fact, are beta strand membrane proteins. The black line here, this one is accuracy or precision, while the line with the open circles is coverage or recall, you see there's a payoff, so you can predict at a high level of precision or accuracy, but you don't find many things. And when you find many things up here, you have a very low level of precision. Now, the first question is, when I now apply that method to running entire genomes, I really have to ask, in order to find beta barrel membrane proteins, I have to define a threshold. So the question is, where do we put the threshold? Which of these lines is the right one? Or is it another one? How do you choose which line you use? You want to analyze the genome. There's actually one more.
I'm not entirely sure, but
Nobody is.
Yeah, but I would say maybe where both of them meet would be a good
Who feels this is a good idea?
point starting.
Who feels this is a bad idea? So whoever feels this is not a bad idea must feel it's a good idea. Or you don't have any better idea, is this definition of this is a good idea. So get used to it.
It all depends on
So anybody feels it's a bad idea? Anybody has an argument against it? So let me tell you, this is what in fact what most, I would argue most publications do this. I believe every student in my lab at some point came and made exactly the same proposition. I don't believe it's a good idea. I don't believe it's a good idea because these two curves have no reality. If these two curves have no reality, where they meet has no reality. But again, in many, many publications, very well-known scientists make exactly that decision. And they make the decision in a world in which I don't have any other argument. This is as good as any other. Okay? And that so is important to remember. If there's nothing else I can say, I believe this is a good choice. But there is, I claim, something else we can say.
it's like I think it all depends on.
Look at the curve. Let's begin with a curve. Just let's iterate from the intersection point. Look at the intersection point. And do you immediately have a sort of epsilon improvement over the intersection point? Yes? Is that a yes or is that?
Well, it gets even better. Like after the intersection.
That one gets better, but that one gets worse.
But that one gets worse, so I think it's kind of always a
But don't go that far. Just stay in the immediate environment.
But for some time the
Exactly.
line is the same.
So the immediate improvement over that, so if you begin with the intersection point, you immediately would argue, but listen, here we have a flat curve, meaning that this one is flat and this one increases. So I can only win. So if I provided my curve is right, but this is always the assumption, I have to trust this curve. In the end I may say, well, I come up with a threshold and now my threshold may not be entirely right. But as long as I go to the threshold, I have to sort of assume that essentially what I see is right. And if I assume that, then at least here is some sort of stable, or some point where coverage is not, or recall is not changing, but precision is. Meaning, moving a little bit right of that, I clearly gain without losing anything. Okay, so that is the first improvement over the intersection point. And that already is why the intersection point here is not the right solution, because you immediately have an argument why you find something that's better than that. But it's still not it. There's still something else you can say. And that is sort of more in the context of what you know about biologists and times to do experiments. Yeah?
It always depends what you want to do. Like, do you want your hits or your predictions, or the positive predictions, to be with a high accuracy to be true predictions, which would be if you have experiments which are very costly or time intensive, or if you can just like do experiments on high throughput and you just want a high coverage, then you could
Okay, so let's just assume, again, that is true. But you're answering, the way you phrase it, you say it in a way that is, let's say, generic. This is a generic truth No matter where if I showed you data from chemistry, physics, or whatever, what you answered essentially is true, right? But how is it really in reality? In biology? How long? Say we have the situation, we run an entire genome. We look at an entire bacterium, and we want to identify new beta barrel membrane proteins. Okay? And you have an experimental collaborator. What do you believe? Without asking them, what do you believe is realistic? How many can they test in a tube?
In what time?
In 3 years, in a PhD project. So this kind of project that I'm showing here, in this particular case, for Henrik it was a PhD project. But today, because we have more labeled data, today that would be easier. And he was the first who did it, but today this would be a master project. And so after a master project, you come to the point where you can run new bacteria that somebody experimentally has to do in a PhD project. In a master project, essentially the answer is give them 0, and then they can do it in a master project. But Pia, Anton?
The obvious answer would be that they want the proteins where I'm sure. So the less and the more sure I am, the better.
Yes. Sorry for the animation. Is that what you wanted to say?
Yeah.
So in fact we picked this point here, the first point, essentially where we believed we were 100%. We know we are not 100%. But this for us was clear. When we know that we make mistakes, that is sort of already wrong. When we believe that we don't make mistakes, most likely we will still make mistakes. But that would be the point to start.
I have a question about this choice. So there is a degree of uncertainty in the graph. So picking the point where it's still at 100% is kind of arbitrary.
Yes, that's true. That's a good point. So, and one way the way I look at these graphs, ultimately, is there must be a smooth line. Yeah. If you had enough data, then there would be a sort of differentiatable line somehow. And if you see that here, then it's unclear where that runs. But what seems to be clear from here is there's some part where you're most likely will be somewhere at 100%. And most likely what you can sort of take home here is that the real line would sort of go somewhere here. So maybe you would move it a little bit to the right. And that is a good point. But then we observed that sort of moving it half to the right. So the drop in recall is from 50% to moving it a little bit more in a safer region. This drop here is so hard that we essentially picked that point. May have been over-optimistic. But now we get to the next issue. So one way of looking at it is the way that I showed you here. Another way of looking at it is what essentially do you do for proteomes? Now what I mean in this is not only when I run an entire proteome, how many membrane proteins do I find? There's something else I have to look at. What is that? So I have my method, I have my threshold here, and I can apply that to E. coli, okay, or to some other bacteria, and see how many do I find. What is it that I have to do now? What else do I have to do? Is it enough if I say in E. coli I find 50? Mind you, the method was set up by just a few handful, in this particular case 15 protein structures from the PDB. Now I run it for E. coli and I cannot remember what the answer is. But what do I have to look up? We need to compare that to what is already known for E. coli. I'm developing a new method to identify new membrane proteins. If the ones that come in my threshold, the highest here, happen to be the ones that are known, my method doesn't help. So if I gave an experimentalist things that are going to give him or her an experiment of finding a beta barrel membrane protein, great, without any mistake. But then if those are, at the end of 3 years, are known, not so great. Well, maybe I argue as a computational biologist at that point I'm living in a different country, so they can't reach me anymore. but that's not quite the kind of angle I wanted to get. Yes, Benjamin.
Yeah, but isn't it, when you say we have to compare what is already known about E. coli, didn't we do that when we set up the model?
No. Where, in what I show you so far, did we do that? So this model here is a model that is done on PDB. This, in fact, there's no other way of doing that. You need structures of proteins where you know exactly, is it a strand, is it a loop, is it an upstrand, is it a downstrand? That's the way you build them all.
Also, the 15 PDB structures are not PDB structures from E. coli. They're just PDB structures for beta.
The entire world of, that's 2004. In 2004, the entire world of all beta barrel proteins in PDB sequence unique was 15. The number is not much higher today.
Really?
Today the number is in the ballpark of, and it certainly is not double.
Information too.
That's very little information. And in fact, this is the entire set. And for this entire set, so Henry really spent the major part of his, or a substantial part of his PhD in labeling them.
I don't get this. So we have 15 structures from which we know that there's a transmembrane protein.
Mm-hmm.
We build our model on that. But we don't have anything else to compare it to. So there are no more structures.
That's not, yes, that is correct. There are no other structures. But that is not the only thing you have. And we are getting there, I'm sorry for the animation. So I'm not going back here. So let me just keep going. I'll address that point more explicitly. So if we run it for a particular organism, one way is essentially to say, well, in that species, I predict that set, that percentage of beta barrel membrane proteins. The other one is to ask, I could have two different thresholds, right? I could try a higher threshold, I could try a lower threshold. This is hypothetical data. But then we sort of, this one would be that threshold, that would be that threshold, so this fraction is the more reliable, this is the higher recall threshold. But the point is, what is missing is what is there already, and that is not only what is in the PDB. So what we currently have is this one. This is a bunch of gram-positive and gram-negative bacteria. And this is, the green part is low resolution experiments. Okay, this is not proteins for which I know exactly which residue is in the membrane, but I do know their barrels experimentally. Okay, and you see already, and this is now number really of proteins in that organism. Haemophilus influenza, E. coli are the first two here. So if you can't read that from the back, so for Haemophilus influenza, this is about 24 proteins for which I experimentally know in Haemophilus influenza that they are beta membrane proteins. Okay, what else do I need to do? By the way, this adds the story of the gram-negative. So beta barrel proteins, gram-negative bacteria have two levels of membrane. They have two membranes really. And beta barrel proteins are on the outside. These here don't have beta barrel membrane proteins. So this experimental knowledge is not known. Okay, there's an experimental knowledge of something that is not true. Doesn't exist. So somebody found that in this bacterium, I don't know what the paper is, there's experimental evidence that this is an outer membrane protein in a bacterium that doesn't have an outer membrane. So, this is a mistake, okay? This is always something that you also have to remember. Experiments can be wrong even on that extreme level. So, but there's something else you have to do. So this is all of the dark green ones here are proteins for which somebody has done the experiment that this is an outer membrane protein. How would you now, what's the cheapest way in which you would find new outer membrane proteins without developing any hidden Markov model?
Sequence sequence alignment?
Yeah, sure. You would run against the set of all experimentally known outer membrane proteins. So the 15 ones in the PDB plus, you see here the numbers are already higher. You would add them up and you would run a PSI-BLAST and see, are there others that are similar to it? Now the story gets a little bit complicated. So how similar do they have to be in order so that you can really say they're beta membrane proteins? So you have to define some threshold. It's true. But if you did that, simple PSI-BLAST run, then you immediately see that actually some very new features come up for these bacteria, right? So that the one that has the highest is no longer the one for which you have the highest knowledge. So E. coli, in fact, is the one with most beta membrane proteins known either experimentally or by homology inference, assuming that the light green here is a very reliable prediction. And then now we get into our prediction method. And our prediction method I show at different thresholds. So the most reliable thresholds here are in fact in blue. So this is an example where I have a very reliable threshold, and it corresponds to a precision of 95 or higher, and the light blue we are going further down in the line. Now the dark blue, so the one that you criticized, Anton, essentially is you said this is maybe not good enough, and it's true, but you also see that for the entire proteomes the dark blue is not seen very often. Also you see that dark blue is at least in one of the proteins that don't have outer membrane proteins. So this is a mistake. The confirmation of the good thing is that there's very little dark blue on the side of bacteria that don't have it. And even the light blues are still very little, while here the light blues in some cases really add up to considerable numbers. So the point of this here is that just the prediction method is not good enough. So you have to also put that into perspective, and in order to produce this slide, we in fact had to also work out in a separate experiment what is the homology-based threshold here, and it's not the typical H value. We used a slightly different story here, and there's a story underneath. But in order to get to that slide, it's not that easy, and then you get a slide that is not complicated, but that's relatively complicated. But this is in fact what you have to do. Now, what we could discuss is whether we should at all show the purple ones here, because the purple ones are really, we have low trust or low reliability. But now we get into the discussion that, and I keep looking for your name, but I can't get it. It's not Anne. You're not Anne.
Maria.
Thank you, Maria. Sorry for that. And I cannot find you under. I'm confused because I should have known. So now we are exactly at the point that you said, Maria. If some experimentalists were to say, we want really a high coverage, we want to find many things, because the light blue is not enough, because I'm working on Aquifax aerobolicus, and there is no blue in this. So give me, in that particular case, give me your red or your light blue. And again, I'm not entirely sure. So 35%. So this is a threshold somewhere here, right? So we're going down to levels like this for the entire protein. And you still don't find many. Now, what we don't know at the end of the day here is whether the numbers are really supposed to differ. And you see a big difference of these bars, right? Is that because these bacteria really have a different set of membrane proteins? We don't know. We have no idea if in the future somebody came up with an experimental way to label them all, you would see the same spectrum, or you would see something that is flat. Meaning that the sum over all of these would be similar, essentially, or proportionally similar between the bacteria. Well, proportionally similar, so the bacteria are also different sizes. Percentage I don't show. Here's one where I go even further down. Okay. Any further question on this issue? 1D solvent accessibility. And now we get into another feature that you can write in a string that is used as input to many other methods, and initially was used to sort of predicting structure. We get it from known structures through the idea of essentially rolling a water ball, a water molecule. And in this particular case, we would say that C and D, residues C and D, are the only ones that are not accessible to this rolling water ball. Because the ball rolling over it will not touch D. It will touch all the other 5 residues, but not D. And we can take this rolling water ball and define a square angstrom surface. Okay? We simply see what surface of the ball or the water molecule reaches the surface of the protein. Okay? And from the way proteins form, you know that some side chains are very short, some side chains are very long. And this means that if you look at these square angstrom solvent accessibility, if you had B as a short side chain in adenine and C would be a lysine, the absolute solvent accessibility for C might be higher than for B, although C is sort of more on the inside of the molecule, right? Because simply C has this long side chain pointing out, for instance, lysine. Now, how could you, if you then began to sort it, so if you wanted to use solvent accessibility to sort how deep in the protein a particular residue is, you would make a mistake because it would sort of sort C to be more outside than B. Okay? How can you measure solvent accessibility in a way in which this cannot happen? Or would not happen that likely? What else could you look at? How could you make the solvent accessibility become independent of the size of the side chain? No idea?
Well, the obvious solution is just to use the atoms and not focus on individual residues, but then again, atoms can be really, really hard to calculate because there's so many.
But then you want to predict for every single atom how exposed to solvent that is? That brings us into your solution is very difficult to pay for on the other end. So now you suddenly have a method that predicts for every single atom in a protein the solvent accessibility. Nobody has ever tried that because it's actually not that easy. We have a lot of states.
So maybe you can just take the length of the chain and then just sort of subtract it or just add it to the accessibility, to the how far outside it is, so subtract it.
The length of the chain, so you take the length of this chain and then what?
Just say, okay, the radius of this amino acid is actually larger because it could be the whole chain.
I believe you're getting, I believe you're thinking about this. That's what you meant? Yeah. So essentially you define, that's exactly the story of the radius if you want. So the way I think about it, you define a relative solvent accessibility that simply says the maximal solvent accessibility is different for this one and that one. It sort of translates by the radius to fit this one is higher. If you had a ball to fit this one, the radius of that ball would be higher than the radius of the ball fitting this one. Okay, and then you're essentially not measuring absolute solvent accessibility, but you're measuring this relative, so what fraction of the maximally accessible surface is accessible. Okay, so that's a percentage value. Now the percentage value, you can convert it to states. In states you have, for instance, two-state. In a two-state model you have residues that are buried in the inside and residues that are exposed. They are the ones that bind. They are the ones that bind to water, but they are also the ones that bind to substrates. They bind to other proteins, they bind to DNA, right? While the ones buried are important to fold and to keep the stability of the protein. And then, you could imagine that there is sort of a three-state system in which you have the ones that are really in the inside, the ones that are really exposed, and somehow the ones that possibly can change by motion. So that could become exposed under some conditions, that are a little bit exposed, and that could become sort of connected to the inside, to the core. Now the question really is, what is best? which one are we going to, so we want to predict, develop a prediction method, which one should we use? Let me make the story even more complicated. When you look at a residue that is 100% solvent accessible, or you look at a residue that is 80% solvent accessible, both of those essentially are solvent accessible. They are somewhere on the surface. And it just has to do a little bit with the rotation of the residue. And that is again the kind of thing that happens in motion. So the same residue in motion can move from being 100% accessible to being 80% or the other way around. So this is not really important. But whether you are 0% accessible, that only happens when you're really inside the core, or 20%, that's a big difference. Because 20% still means that there is some access to water. That you are at least binding a residue that is close to the surface. And that even some of this residue that is 20% accessible, even some of that is accessible to water, right? And that is different from the ones that are really in the inside core of the protein, okay? So on the one end of the spectrum, the difference of 20 percentage points is important. In the other end of the spectrum, the difference is not important. How can we reflect that? Yeah?
You use the logarithm for it. Because
You can use a simple function. Logarithm is one idea. The solution here is the square root. The square root of the number, of the integer number here, gives the relative solvent accessibility. And essentially, so you square 1, 2 to 4, 3, 9 to 16, 16 to 25, and so forth and so on. So, historically, the way we predict solvent accessibility is by hydrophobicity. Again, residues that are hydrophobic are on the inside, residues that are hydrophilic are on the outside. More recent, again, on your scale, this is not really recent, it was done 20 years, 22 years ago, using a neural network. Essentially, it's the same machinery that was used to predict secondary structure, using profiles as input, plus all kinds of other information. And this particular neural network had 10 output units. So where each of these units stands, the number of the units, so unit 1, 2, 3, 4, 10, stands for the square of that number in terms of relative solvent accessibility. And the idea is clear, right? The reason why do you believe I took 10? Why not 2 for buried exposed? Or why not one number for relative solvent accessibility using this function here? So you could have done that too, right? Why not? Why do you believe I didn't do that? In the words of Simon in the part before, you simply try them all and you see what works best, right? And this is exactly what I did. And so this worked better, plus it had the additional advantage that it actually predicts relative solvent accessibility not in a binary way, so buried exposed or buried intermediate exposed, but it leaves it to the user to choose if they want to change. So this comes also with a bunch of models. So it also is a prediction is buried versus exposed. But the users may, according to their interest, change because what they get is the actual number. And this I can also translate back into a square angstrom solvent accessibility. So users can from that number get anything, and it works most reliable for predicting relative solvent accessibility. Now here is again the idea of the reliability index. Again, so I'm not going to show you the 10-state model. I'm going to show you an example for the 2-state model where I've buried exposed. And say the unit of buried has an 0.9 predicted and the exposed has 0.1. Then I predict this to be buried. And if this number would be 0.6 and this one would be 0.4, I would also predict buried. Both of those are predictions for buried, but this prediction here is less strong than that prediction. The distance between these two numbers here is higher than here. Now, when I predict for every single residue, I simply say, well, I take whatever is the largest. I take the unit that is predicted to be the largest. And that is the point with the open circles here. For that, in a two-state model, I get 74. When I am allowed to return only the subset of predictions for which the threshold is larger than some number, which happens to be on an integer scale 4, then what I do is I look. 4 here is about 50% of all the residues. So that means when I require that the distance is larger than 4, 50% of all the residues are left to predict. All the others on that side here are residues where the difference is less than 4. So I don't predict for those. I'm not predicting either B or E. Just don't predict. And for the ones for which I do predict, again, half of all the residues, the performance here is 84. Right? Or you could also put yourself on, say, the 10% strongest predictions, for which the distance is higher than 9 on that scale. And for those you would have an expected accuracy of about 90-something%. Okay? The same is here true for secondary structure prediction. You have also an increase. Again, this is a 3-state model. This is a 2-state model. So you can, in any prediction, this axis in some sense is given. You know for every single sequence, you don't know anything about it, but you can predict solvent accessibility, and you know what fraction of your residues are predicted strong. At the threshold above 9, you can look up in your plotting. What accuracy there is, you can only know for the data set that you initially have to assess the performance, right? And then you provide the user with a set and you tell them, listen, when you look at the 10% strongest, you expect that the accuracy for those, or precision for those, will be 92% or 91%. Okay? The idea of reliability index is clear? Was it clear before? What else do we have to do to make it clear in an exam? Will it be clear in an exam? Good. Actually, you guys came with that package, right? That's one of the questions. Anyway, good. And by the way, this is for membrane proteins, but this is per residue scale. So this is not really that relevant. Now, detailed prediction, meaning what percentage is correctly predicted, or what percentage solvent accessibility is predicted, is that correct, is more problematic. And that is more problematic because that number, percentage solvent accessibility, is in fact not very well conserved between homologs. But if we sort of go into a more coarse-grained model where we have a two-state, so what fraction is buried, what fraction is exposed, and typical values to distinguish between one or the other is 16%. So less than 16% is called buried, more than 16% is called exposed. At that level, about half of the residues are buried, half of them are exposed. And at that level, levels above 75% accuracy are reached. So in this particular case, the training goes to maximally 80%. Why? So I can completely overtrain my model, and I still don't reach much more than 80%. Again, on a completely new data set, I reach 75, so I'm not that far away from that. Why do you believe that's the case? Ultimately, this relates to the issue of the evolutionary conservation of solvent accessibility, which is much less than the one for secondary structure. So this is the percentage 3-state accuracy of conservation, and this is the 2-state of of secondary structure, and this is for solvent accessibility. And on this axis here is percentage sequence identity. That's not really important, but overall what you see is that these bars, random for these here is 33, random for those is 22. Nevertheless, these bars are substantially higher. And again, I point out to you, this is the same scale we are looking at. So the blue bars are clearly higher. Secondary structure is much more conserved. for proteins that are very similar, so that's sort of roughly the H-val, and for proteins that are more distant in the twilight zone. For all of them, secondary structure is more conserved than solvent accessibility, and ultimately that is, by the way, the green one here shows that is the number of points, so most of the pairs that entered into these graphs are actually sitting here. Are sitting here on above this line. So counting below here is there is still significant numbers. So there are many thousand pairs above, below here, but most of the data is sitting up there. So that ultimately is the problem. So red is less than blue. Solvent accessibility is less conserved than secondary structures. Ultimately, the problem why we cannot quite train it. What we could do is, in fact, redefine the problem and say, define solvent accessibility such that we find a measure which is not quite solvent accessibility, but something that reflects where on the surface you are, whether you're binding or not, that relates, or that is a feature that is more conserved. And this is an idea that some people have addressed. There are some solutions for that, and it's an idea that I believe is about to be picked up again. There has not been done much over the last few years on this. So what do we get out from these predictions? One example, and this is not from a prediction, but is more a generic example how you can use accessibility over the surface, a tool from the group of Nirbental, whose name is not on the slide. That is that paper from Fabian Glasser. Conserve. So you take a protein surface and put a color onto it according to what is conserved in an alignment. The more conserved, the more red. The less conserved, the more cyan here, right? Or the more blue. And looking at this, you may immediately sort of see that you have a binding site. It looks like a hole that is conserved, most likely is a binding site. Now, looking at the molecule without the colors, you would not immediately see that because you see this kind of dents or holes in proteins all the time, and they're not all binding sites. But since you see it's evolutionarily conserved, you immediately know how to interpret it. And by the way, this tool conserved surface is one of the best ones. You have a structure to one of the first things you should in which you should combine visually knowledge of structure with knowledge of an alignment to get some idea about its function. But here we have an example for a cavity that in fact is not conserved. Cavities are not always conserved. This is an example for that. Sometimes conserved cavities may bind to different substrates. So from knowing the cavity, you're not quite sure what it binds. So just making something fit in there is in fact not always working. And at that point, I believe you need also a different view. You need the view of electrostatics. So at that point, you would possibly, would most likely also want to look at it from a sort of more energy perspective and color according to biophysical features. Here you color only according to conservation, meaning importance. So that's the first step, to identify the sites you want to look at. What it binds to is the next step. But now getting back to, and the conserve method can also be used for proteins for which you do not know the surface, and then you essentially use solvent accessibility predictions to get at the surface and color the surface residues that are conserved, and do the same thing. Then you cannot no longer make the nice pictures though. Solvent accessibility helps in the prediction of subcellular localization, protein-protein interaction, and the prediction of motion or flexibility from structure. B values is one story that we will not get to in this semester. Now, these are features, these were the first features for which it helped. Many others have been added since. But now I want to look at a completely different way of using it. And the different way of using it gets back to this idea of domains. A domain is a compact three-dimensional unit. It's something you take, the idea of a domain, there's no definition of a structural domain. But the idea of a structural domain is you cut a structural domain out of a sequence, you put that sequence into a solvent, it adopts a unique three-dimensional structure on its own. And that essentially is a domain. Okay? If you sort of cut a fraction of a domain here, this would be one idea, then it would not adopt 3D. It would not fold. Okay? Or would fold in different ways, misfold. Then there is this idea of a chain. A chain really is a domain plus a little bit. So the whole protein may not be only the domain. It may have sort of an N-terminal and a C-terminal loop, and there may be some other additional loop somewhere, some other addition, embellishment if you want. But then the major domain is sort of the thing that many other proteins have in common, and maybe some of them here have a signal peptide. And some of them have some other reason to a C-terminal peptide to retain your protein in the endoplasmic reticulum or something like that. And then there are regions that are sort of fragments of a chain. So they clearly are not the domain, they clearly will not fold. So now the idea is that we take a sequence, predicted solvent accessibility, and use that to see on which of these sites we are. So are we looking like a domain, or are we looking like something that is a fragment of a domain? How would you do that? What do we need in order to do that? So I have a method that predicts buried versus exposed residues. How can I use that to predict domains? Okay. Yes?
If domains are supposed to be more globular, then it should be sort of like bubble of...
Yeah, but you don't see bubble. I have a residue. I predict whether that residue is B or E. And I do that for all 100 residues in the protein. How do I see a bubble in that? Essentially, don't forget, we're talking 1D features. So far, I showed you 3D pictures, but what we're actually predicting is 1D. I have a string. Yes, this is a nice image. But how do I put this string onto a bubble?
So I guess I would iteratively go over areas that are between exposed residues and then enlarge them, with the idea being that if a residue is exposed, everything that comes from one exposed residue to next exposed residue will be inside the protein. And so this is like my first domain, and then in the end a domain will be everything that is from the first exposed residue to the last exposed residue in my sequence.
Are you saying that you essentially want to sort of make a frequency plot of the length of buried regions?
If you will, yeah, that's because...
that idea never occurred to me. It's an interesting idea. I never thought about that. I don't know whether that would work. Cannot tell you.
But that's like the first idea, because...
No, it was not my first. It was not my first. I never had it. I only took my first idea and never saw anything else. And that was not the one.
I have an issue with your idea because it can be buried, but it can be like the loop, so it's exposed.
We would have
buried, it goes...
Gary, this is true, but we have to... So the only way to find out whether that will work is we have to plot it. I haven't done it. I have no idea.
Actually, I don't know how you want us to know how, only from knowing if something is exposed or not, how to derive a domain.
Really? Don't think about it.
There's certainly more information than zero, so you can make some.
So, okay, think. Now, imagine a protein were a cube, right? And is made up of little cubes. Each amino acid is a little cube, and you build up a protein from these little cubes. Then you can clearly relate the number of cubes on the outside to the number of cubes in the inside when you just have a single number of residues. So you know for a certain number of cubes, you have a certain ratio, right? That's very easy to plot. So the next thing that you can sort of improve from that is you assume it's not a cube, it's a sphere, okay? And those are the two situations that I showed. Here's the protein length, number of exposed residues for the assumption that we have cubes, and for the assumption that we have spheres. And these two you can program immediately. So you can figure out the formula for that in your own head. You don't even need to Google. This was done at the time that Google didn't really exist. So this is the kind of thing you can easily do. You're all fit to do something like that. And then you simply look at the observed data in blue. And what I found, I mean, I know that the protein is not a sphere, and I know that all amino acids are not the same size. So this is an astonishingly good fit for such a trivial, simple model, which I know is wrong, right? The blue, and just assuming that we don't have a sphere, but we have an elongated sphere, the equation gets a little bit more complicated, but it moves in that direction and you go closer to the fit of the data. But if you have that, then you can simply argue, well, I have my fragment and either fit to this or I don't. You could also, I mean, the actual refinement was done by fitting the curve, but this is just showing you how remarkably simple the fit actually is, how remarkably similar proteins are to cubes, or at least to spheres. Yeah?
I have a question. How can you figure out if you have more than one domain? Because probably if you take only one domain proteins, I guess this will be pretty good approximation. But if you have many domain proteins?
Yes, so that is totally true. So this works for single domain proteins much better. Now if you have a two domain protein, you're not that far away from it, because essentially you have two dishes of the same thing, and you sort of could imagine that yes, it works best for single domain proteins. That's true. Now here is a data set. What I wanted to apply that for, what I applied that for in this graph, was when yeast was first sequenced in '95. 9,000 proteins were published. Okay, that was day one. Do you know how many proteins there are in yeast today? Any idea? Anybody? Yeast has. Do you all know how many we people have? We have how many proteins?
20,000.
Is that difficult for everybody? Is that not everybody knew that? Okay, so we have 20,000 proteins. Yeast have 6,000, sort of 600 roughly. On the first day, 10,000 were published. On the second day, there were 6,600 on the website. What had happened in between is that the people who had published the first website simply had retracted 3,000 something genes, and they had retracted them because they were all very short. They were less than 100 residues. And for shotgun sequencing, if you're too short, the error rate goes up. In order to clean up the data, they had essentially removed all the short proteins. Now the short proteins are in fact the ones that are single domain. So the method initially was meant for short things. Anyway, so what we have here is the distribution for what was believed to be the yeast proteins. What we have here is globular proteins, globular domains, and this was sort of the fragments that were removed. And ultimately, the statement was that it allows you, this is the red one here is a curve summing up to 3,000 something, 3,300, and you can at least find, 30% of these 3,000 or 25%. So there's several, maybe there is 700, maybe there's 1,000 in here where you can say this clearly looks like a globular protein, and this most likely is not a mistake. Because we know that several of these here in fact are known proteins and have come back into the database ever since. And this was simply an idea how you can, with a very, very simple method, sort of find some of the ones that previously were thrown away. So there are other 1D related or 1D features related to solvent accessibility. One is the OI number. That is essentially the number of C alpha in a sphere. And again, this is something that people have tried to predict. Another idea, I briefly mentioned that from Rachel Karshan, that comes from Kevin Karplas originally, was the idea to really choose what you measure as a solvent accessibility according to what you can predict best. And that is in fact the publication in which they did that.